screen. Which screen are you seeing?
Yeah, the right one.
The right one? Wonderful.
Yeah.
Again, hey everyone, thank you again for Katie Ebru and Esther Lally in organizing this amazing seminar series which bring us together during these challenging times of pandemic. And I also appreciate everyone tuning in for the seminar today. I wish everyone family well. Like Katie said, our lab is in the last almost 2 decades or so studying the phenotype of innate immune memory dynamics and how they relate with disease such as atherosclerosis. For this group of audience, I think there is no need for too much introduction about the significance of innate immune memory. We recognize that innate immune cells, they're not just simple executioners of first line of defense, but rather they can be programmed into diverse repertoires of pro or anti-inflammatory cells with distinct features that are closely involved in diverse pathological processes ranging from tissue repair to tissue damage that can be reflected in almost every aspect of atherosclerosis. Not only in atherosclerosis, but also in other bigger aspects, for example, in the current climate of COVID-19, that the dynamic changes in innate immune cells clearly play a role in the pathogenesis of infection as well. So better understanding of innate immune memory dynamics can help us perhaps complement the vaccine approach and provide independent therapeutic strategies in treating infection and disease. So before I go into the details about this innate immune memory dynamics, I would first like to, let's see how, okay, first like to acknowledge the people in the lab. We have a group that involve research scientists, postdocs, graduate students, and also undergraduate students. In particular, I'd like to highlight some of the work done by research scientists Geng Shuo and Zhang Yao, who are primarily involved in in vivo studies, and also graduate students Alison Race and Keisha Pradhan, who are involved in some of the mechanistic studies in vitro. So in terms of innate immune memory, in particular, we're interested in this dose-dependent memory. So as you can see that there are several systems in the field that study the innate immune memory dynamics. In the earlier days, people treat the cells with different stimulants. For example, we can treat the cells with either interferon gamma that are driving inflammatory response, or you can treat the cells with IL-4, and then you can polarize the cell into a state so-called M2 state. And so basically, it's depending on the nature of the stimulant, you can artificially drive the cells into different activation state. And later on, people realized that the cells can also adopt some rudimentary memory state. For example, you can treat the cells with beta-glucan first, interferon gamma first, and then you can come back with a second stimulant. And then the cells will no longer be naive anymore, but rather they can memorize the first challenge. They can be trained, if you will, so that they can be more responsive to LPS challenge. In addition to that, our group in the past have been primarily focusing on a different aspect of innate immune memory. Namely, we believe that innate immune cells can not only recognize the nature of the stimulant, but also the signal strength and duration and history of the stimulant. So we use a specific example of endotoxin, the bacterial lipopolysaccharide. So the innate immune cells can recognize LPS, of course, through the toll-like receptor, in particular toll-like receptor 4, but we can also recognize the signal strength. Depending upon the signal strength of the LPS, the cells can either polarize to inflammatory or anti-inflammatory state. They can also be programmed into either prime state or tolerant state or an exhausted state. So, and we study this phenomenon through a combined approach of experimental and computational, as well as in vivo studies. So this is an earlier work we published to illustrate the point. So basically, there's, even with the signal strength dependent programming dynamics, there's two different systems. One system is that you can give a sequential challenge, the first dose, and then come up with a second dose, right? So if we hire the first dose, come back with a second dose, and the cell become tolerant. The tolerance primarily is manifested by the reduced expression of pro-inflammatory cytokines. But then if the first dose is much lower, for example, picogram per ml range, which is reminiscent of the pathological range, in circulation, the endotoxin dosage is never rise above 100 nanogram per ml. Even in sepsis, the endotoxin levels in circulation is about 100 nanogram per ml. But in regular people, in chronic disease, due to the leakage, gut leakage, the endotoxin level is only picogram per ml in humans. So with that subclinical endotoxemia, then we will not get endotoxin tolerance. And there's a second system where we study this memory dynamic. That is, instead of using 2 sequential doses, we use the same dose, but consistently, repetitively stimulate the cell, almost like in T cells, where they challenge with antigen repetitively, inducing tolerance or exhaustion. So we use the similar concept and translate it in innate immune cells, as shown here. So this is a murine bone marrow monocyte cultured for 5 days in the presence of CSF and repeatedly stimulated with rising dosage of endotoxin. Of course, if you stimulate them only 1 dose, the higher dose of LPS, the higher the expression of pro-inflammatory cytokines. However, if we stimulate them repetitively with very, very high dose LPS, for example, 10 nanogram per ml or 1 microgram per ml for 5 days, and then we basically see a suppression of inflammatory mediators that's represented by CCR5 or IL-12. In stark contrast, if we stimulate with super low dose endotoxin like 100 picogram per ml, reminiscent of metabolic endotoxemia in circulation, we drive a low-grade inflammatory state. You can see that there's a robust induction of CCR5 and IL-12. Right? So this is totally opposite as compared with persistent stimulation with high-dose endotoxin. On the other hand, if you look at arginase 1, so with repetitive stimulation of high-dose endotoxin, we drive the induction of arginase 1. So you can never tolerize arginase 1. Neither can you tolerize INOS. Right? So INOS, arginase 1, the higher the endotoxin, the longer you treat them, the stronger the induction. In contrast, the super low dose endotoxin will fail to induce either arginase 1 nor INOS. Okay, so this is the phenotype we saw repetitively, not only in murine cells, using conventional approach, but also we can use the population levels using flow cytometry, looking at the population of cells that can be programmed into one state or the other. Because you can see here, similarly, with lower dose endotoxin, rising dose endotoxin, we expand the cells that express IL-12, but then higher dosages, we reduce the population of cells that express IL-12. But INOS expressing population will keep increasing. And then we also saw that not only in murine cells, but also in human primary blood monocyte. So this is the healthy donors human blood monocyte. As you can see here, as classified by 3 populations, CD14, based on CD14 and CD16. There's a classical monocyte, CD14 high. There's a non-classical monocyte, CD16 high. And then the intermediate monocyte, CD14, CD16. And the intermediate monocyte is often linked with the inflammatory state, in particular in atherosclerosis. As you can see that human monocytes are much more sensitive to endotoxin, and with a very super low dose endotoxin, we can expand the intermediate monocyte. In contrast, with higher dose endotoxin, we completely annihilated the intermediate monocyte population. So how do we explain that? So I term that the sensing of endotoxin by the toll receptors, I call that structural sensing. So basically, we can memorize these, recognize these external, internal challenges based on the different type of toll receptors. However, the structural sensing itself is not sufficient to sense the signal strength and history, and I call that functional sensing. So so that therefore the cells can sense not only the structure, but also the dosage and history of LPS challenge. And that cannot depend only on the Toll receptors, but rather we believe that it depend upon a closely wired intracellular molecular circuitry with unique topologies. And that enables the cells to differentiate and activate depending upon the signal strength. And I'll present some of the data that we collect in the last decade or so. But before that, I just want to summarize in terms of the basic concept. So we believe that every single cell, in order to decide whether to activate in pro or anti-inflammatory state, every single cell, there's multiple competing circuits. They're antagonizing with each other. It's almost like a check and balances. At the same time, they're all responding to the same signal. But depending upon the signal strength, one player will win over the other. And that, as a consequence, they drive a stable and clear activation to one state over the other. But sometimes they can play even. If that's the case, there will be numerous intermediate states as a consequence. So we can run the dynamics. We can see basically that I can explain, even with a simple motif, we can explain multiple repertoires of differentiation or activation state based on this existence of these competing check and balances within the cell. And that check and balances happen at multiple levels. It happens at the receptor levels. For example, there are two adaptive molecules downstream of toll-like receptors. One is MYD88, the other is TCAM2 or TRAM. And we have data collecting showing that superlow-dose LPS primarily go through TRAM instead of MYD88 and drive and then compete. Through this competition, this branch wins over and then polarize to a low-grade inflammatory state. And downstream, you can see there's multiple layers of these unique signatures, key signatures. At the signaling level, we have this MAP kinases, P38, antagonizing ERK. Even at subcellular levels, we have autolysosome either fusion or autolysosome fission. Higher dose LPS favors autophagy, favors fusion. Superlow dose LPS favors fission, disruption of the fusion process. And then that relates the signal to transcription factors that can compete with each other as well. Just want to go through some of the data to illustrate the point I just mentioned. So for example, if you look at AKT, it's been well known, widely studied with higher dose LPS, we see a robust induction of AKT. Drives ERK, drives the homeostasis, cell proliferation, survival. In contrast, with superlow dose LPS, we see a sharp suppression of the residual AKT phosphorylation. That happens also with ERK. Higher dose LPS drive up ERK, but superlow dose LPS suppress ERK. At transcription factor levels, for example, IRF5 is a key typical transcription factor involved in the M1 inflammatory monocyte polarization. High dose LPS, persistent stimulation with high dose LPS almost completely destroyed IRF5. With superlow dose LPS, is totally opposite. It's actually an induction of IRF5. Not only happens at this level, but also at subcellular level. We zoom in, looking at the autolysosome fusion. In resting cell, naive cells, they can have constant fusion between the lysosome, as labeled as LAMP1, and autophagosome, labeled LC3. You can see the overlay fusion of autolysosome. However, if we stimulate the cells with superlow dose LPS persistently, and we polarize the cell that exhibit a phenotype of disruption, you see the disruption of fusion of autolysosome and phagosome. So, and recently we also saw that because of the disruption process, because of the disruption of autolysosome fusion that accumulate some of the molecules, for example, P62, MLKOL, and also IKK beta. So with the superlow dose endotoxin stimulation, we see the accumulation of this one due to the disruption of autolysosome fusion. As a consequence, that drive up the NF-kappa B activation. In the meantime, because of the disruption of autolysosome fusion, we also accumulate the suppressor of NRF2, KEAP1. Right? So as it has shown here, it's been known in the past, high dose LPS drive down KEAP1, allowing NRF2 to activate. So, and then, but super low dose LPS, in contrast, accumulate KEAP1, therefore suppressing NRF2, and then suppressing not only drive NF-κB, but also suppress NRF2. We looked at the flow cytometry. Using flow cytometry, we also saw super low dose LPS expand LIC population. And then if we zoom into the LIC population, we also saw that KEAP1 is enriched. So LPS also enhanced KEAP1 expression within the LIC positive population as well. We further show that indeed the super low dose effect is dependent upon TRAM, not MYD88. If we use MYD88 dependent cells to see the similar analysis, 5-day culture, we saw the similar dynamics in terms of CCR5 or IL-12. Basically, higher dose LPS suppresses, tolerizes CCR5, IL-12. A super low dose LPS primes, polarized the induction of CCR5, IL-12. In contrast, that effect is abolished in TRIF knockout or TRAM knockout monocyte. And we further look at this accumulation of P62 in these cells. For example, look at the P62 super low dose LPS induces P62 in wild type cells, but if we use a TRAM or TCAM2 knockout monocyte, we saw the LPS completely failed to induce P62 accumulation. Neither the LPS can accumulate KEAP1 in TCAM2 knockout cells as well. And if you use the TCAM2 knockout or TRAM knockout monocyte to do this 5-day culture, the programming using super low dose LPS, repeated stimulation, the LIC population almost completely disappeared. So there's no induction of LIC populations if you not use the TRAM knockout monocyte. And we also recently did the single cell sequencing analysis, as you're shown here, that the PBS monocyte primarily cluster together in a naive state. Of course, with the cells repeatedly stimulated with 5 days of LPS, super low dose LPS, they are largely adopting a pro-inflammatory state, as shown here. However, like I said, because the cells, the advantage of doing single cell analysis is that because there's always heterogeneity, then some cell may win over. So between these check and balances, the other arm might win over in certain cells. Therefore, indeed, we can see that feature with single cells analysis that some population cells are becoming tolerized or even exhausted. And I'll show that in the future. And indeed, that fundamental features of check and balances, mutually inhibitory motif, is not only evident in the activation and polarization of mature monocyte, but also in other generalized areas of cell differentiation. So for example, this is reviews by Alan Rothenberg and also Friedman, showing that during the early differentiation processes of myeloid cells and lymphoid cells, you see these key signatures of mutually inhibitory motif between PO1 and NOS signaling that drive a clear decision-making. Even from the hematopoietic stem cells, each step along the way that drive the clear decision-making of one state versus the other, we have these mutually suppressive motifs that make sure we have a clear differentiation. Just to summarize what I just talked in the first half of my seminar, we believe that we found that innate leukocytes can indeed adopt memory state, not only depend upon the nature of the signal, but also depend upon the signal strength and history. And we also show that the subclinical super low dose endotoxin primarily polarizes a low-grade inflammatory monocyte state, highly dependent upon the TRAM or TCAM2 pathway. And then that bears relevance in atherosclerosis. So this is the work we published several years ago, showing that if we chronically inject subclinical dose endotoxin, that is almost 5 nanogram per kilogram. It's about 100 picogram per mole. So this is almost 1,000-fold lower than the low dose that people usually inject. Right? So when people talk about traditionally low dose, they use 1 microgram per kilogram or 5 microgram per kilogram. That they call the low dose, right? So so the super low dose we inject is 5 nanogram per kilogram. So you convert to about 100 picogram per mole. So that can drive this significant increase in the lipid deposition, but more importantly, drive the destabilization of the plaque. So we see a large increase in necrosis area, reduced collagen deposition, reduced smooth muscle actin deposition in the plaque, and also significantly drive the recruitment, drive this deposition of macrophages within the plaque as well. If you further look at the nature of the macrophage within the plaque, we can also saw they are polarized into inflammatory nature. So you can see that macrophage co-staining, they significantly increase the SRA, pro-inflammatory SRA, or reduces SRB in the plaque macrophage. Just to summarize, so basically what we saw is that this subclinical endotoxemia can polarize low-grade inflammatory monocytes in vivo, and that is correlated with the pathogenesis of atherosclerosis. And then I use the next couple of minutes to talk about some of the work we've done in neutrophils. And we collected data showing that this polarization and memory dynamics by endotoxin is not only applicable to monocyte, but also to neutrophils as well. So if you inject subclinical dose endotoxin repeatedly to mice, and if we collect the blood and also collect the spleen, aorta, we saw a significant expansion of neutrophil populations. So this is in circulation. We see expansion of neutrophil populations in blood in mice injected with endotoxin. But also, if you look at the neutrophil polarization state, right, look at the markers of neutrophils, we see inflammatory polarization. So for example, we look at CD11B, there's the induction of CD11B and dectin-1 on neutrophils collected from mice injected with superload endotoxin. But if you look at other markers, anti-inflammatory markers, for example, if you look at anti-inflammatory LRRC32, FPNs, LPS suppress the anti-inflammatory markers. So polarize it to a low-grade inflammatory state in neutrophils as well. And we look at the we also look at other markers. For example, neutrophils, they're the potent producers of inflammatory mediators like MMP9, leukotriene B4s, those are lipid mediators for inflammation, MPO, causing tissue destruction. And indeed, superlow-dose LPS can drive the induction of MMP9, leukotriene B4, and MPO as well. And further, superlow-dose endotoxin can synergize with OSLDL. OSLDL itself can also drive similar production of MMP9 leukotriene B4, because OSLDL has also been shown to partially signal through toll-like receptors. But if you put them together, we can see further synergy between these two. And then also for the cell surface markers, C11B, dectin-1, there's a synergy between OSLDL and LPS. And furthermore, we look at one of these oxidized CAMK2 is induced by LPS, 5-LOX lipoxygenase, responsible for the production of leukotriene B4, also induced further synergized between LPS and OSLDL. And likewise, the key transcription factor STAT1 can be stimulated by LPS and OSLDL. And we also saw, due to this increase in OS-CAMK2, we also saw there's an increase of intracellular ROS by superlow-dose LPS, and also synergized by OSLDL as well. The neutrophil is unique because neutrophil has relatively few mitochondrial content. So the mitochondrial ROS, we didn't see much of a difference because of the lack of very few mitochondria in neutrophils. And also we don't see much of a difference in terms of oxidative burst. So therefore we look at another contributor for ROS in neutrophils, in particular in peroxisome, what we saw is that here is a typical neutrophil as represented here with multi-lobe nucleus. You can see the multi-lobe nucleus here. And in the naive neutrophil culture, overnight, we see there's a constant homeostasis of peroxisome, as shown by PMP70, co-staying with lysosome. There's a paxophagy happening. There's a turnover of damaged peroxisome. However, if we treat with superlow dose endotoxin, we disrupt the peroxisome homeostasis, disrupt the paxophagy process. And we believe that's one of the reasons that superlow dose LPS drive up intracellular RS accumulation in neutrophils. And to test that, we use a compound called 4-PBA. 4-PBA is a phenylbutyric acid. It's a derivative of butyric acid. It's commonly known. I know Ebru has done some nice work in the past. So commonly known to be present in commensal bacteria. Also, we can generate that through exercise. But this phenylbutyric acid is a general reliever of cell stress. And recently been shown is a potent stimulator of peroxisome homeostasis in generation of peroxisome and drive the induction of multiple PAX molecules in facilitating peroxisome homeostasis. So indeed, if we add 4-PBA together with LPS, we restore the fusion of peroxisome and lysosome. And we look at RS, also the 4-PBA can robustly reduce the RS production in neutrophils by LPS. And we also look at downstream effect. Indeed, the induction of OX-CAMK2 and 5-LOX by LPS can be potently suppressed by the addition of 4-PBA. And so does the STAT1 activation can be suppressed. And look at the phenotypic markers, the induction of MPO, leukotriene B4, Dectin-1 can all be suppressed by the addition of 4-PBA. And on the other hand, 4-PBA not only suppress the inflammatory polarization of neutrophil, but also can potently induce the induction of anti-inflammatory mediators. For example, LRC32, FPN, recently showed that 4-PBA can also induce the resolvins like resolvin D and others. So 4-PBA can polarize neutrophils into a resolving state. And then we use the neutrophils trained with 4-PBA in vitro, together compared with the PBS program neutrophils. And then we transfuse these neutrophils, about 2 million neutrophils in 200 microliters. We do that once a week, weekly transfusion for months. And that is sufficient to robustly reduce the plaque size and also the lipid deposition. So we believe that the resolving neutrophils trained by 4-PBA can be potently utilized to treat atherosclerosis. And with that, I'll summarize what I just said, that the subclinical endotoxemia can polarize inflammatory neutrophils, synergize with OSLDL, potentially through disrupting paxophagy, inducing intracellular ROS. And that inflammatory neutrophils can exacerbate atherosclerosis. And in stark contrast, if we use resolving neutrophils programmed with 4-PBA, can reduce ROS, restoring paxophagy that can be potently used to treat atherosclerosis. And with that, I thank your attention. This is our team and our collaborators. And again, thank you for your attention.
Okay, excellent. Thank you, Liwu. And as per usual, we're going to ask anybody to raise their hand using the raise hand function if you have any questions. So maybe I'll start. Sure. So I really think that the idea of the leaky gut syndrome that you sort of are mimicking is very relevant, obviously, and is likely to explain a lot of what we see when it comes to exacerbated inflammation. I'm wondering if you've looked at other types of inflammatory, whether it be bacterial or viral or even autoimmune, in terms of maybe not the massive autoimmune diseases that we know, but others that might be sort of chronic, even some of the arthritis, the more mild arthritis, those kinds of things. Have you looked at those responses with respect specifically to the innate immune system and atherosclerosis?
Yeah, that's a great question, Katie. We co-published a paper together with another group, and they're showing that in lupus, in human patients in lupus, and also in animal models, that they have elevated endotoxin levels in that disease model. So I think it's potentially generally applicable. So people also found in HIV patients, they also have elevated endotoxin levels in their plasma as well.
So it's still sort of it's autoimmune or other inflammatory diseases still inducing a basal level of endotoxin, you think is still the mechanism, as opposed to other types of potential inflammatory stimuli, like other ligands that might be inflammatory, or do you think it all kind of comes back to some kind of endotoxin?
Well, I think both can be true. Both can be contributing. I suspect that we also tested. I didn't show here. We also tested some other toll-like agonists. For example, TR7 agonist. We saw the similar patterns of priming tolerance. Low dose TR7 agonist that polarize, and higher dose TR7 agonist tolerizes. But we saw that TR3 agonist that cannot tolerize. They just keep on priming it. So clearly that can be potentially applicable to other agonists, perhaps in different windows, different threshold. But I think this key dynamic motif will hold true.
Okay, Esther, you have a question?
Yes, Esther.
I also have a question. I cannot raise my hand, but
that's fine.
Fascinating story, Liu.
Oh, thank you.
I'm especially fascinated by the neutrophils because they are very short-lived, more short-lived than the monocytes. So how do they keep this innate memory? Is there some epigenetic changes in their precursors, or do they just react very fast, or how does it work?
Yeah, that is a great question. So we also did some preliminary study looking at the survival and longevities in both in vitro and in vivo. So I think even with human neutrophils, if we give them some GCSF or GM-CSF coupled with some TR agonist, for example, very low dose LPS, their lifespan actually expands. So they can survive. They can survive even human neutrophils, a couple of days, no problem. 3 or 4 days, no problem. So in vivo, they can survive on average 5 days. That's why we choose this regimen of weekly injection. Mm-hmm. But in vitro culture, doing the mechanism, we just do that 1 day or 2 days. Look at the mechanism. But you also mentioned a very interesting question, that is, how do we maintain it in the long term? I think that question also applies to monocyte. So even with monocyte, they can survive perhaps 15 days, a month, but still they'll die, right? So in acquired immune response, they propagate through clonal expansion, through this VDG recombination, they proliferate. But innate immune cells, we're testing the theory that perhaps they can propagate through communications. So we think they can passage that through talking with neighboring monocyte neutrophils.
Okay, cool.
Okay, Shui, go ahead.
Okay, hi, Liu, great talk. Yeah, I have a question. About the difference between the high dose LPS treated cell and also the super low dose, which is monocyte or macrophage. Mm-hmm. So we know the high dose LPS is going to induce a titinate production. So then it's going to activate the NRF2. So I just wonder, what is the difference between especially the metabolic regulation and also in terms of the itaconate production?
Yeah, that's a great question. So we just start to look into this with high dose LPS. So clearly, the persistent stimuli with a long time, they actually reduce mitochondrial function. So you look at respiration mitochondria, just completely suppressed it. Super low dose, we don't see any major difference in respiration mitochondria. I suspect that so we haven't looked at the metabolite yet. So I suspect that the effect may be not that drastic.
Okay, thank you.
Okay, Yuri, go ahead.
I have a similar question. Very interesting talk. How do you explain the difference between the mechanism of low and high dose? For example, did you look at the rate of endocytosis of TLR4
Yeah. So that's a great question, Yuri. So Alex Hoffman's group at California, they published a couple of papers in the past showing, so of course they're using a cell line model, showing that the MYD80 response is more transient, more robust, and the TREM response is more persistent, more prolonged. So somewhat complement our studies. And indeed, we haven't looked very in detail about the cell membrane trafficking, but we have some clues. So for example, we use a cell line that no longer can traffic TLR4 receptors from cell membrane to endosome. So there is a GP96 deficient cells that we collaborate with another group. We use that one. We give a super low-dose LPS. We no longer see the effect. So we suspect perhaps the super low-dose LPS must internalize inside the cell in order to signal. And we use the same cell, we use a high-dose LPS, we can still have some response.
Okay.
Yes.
Oh, sorry, Ebru, I didn't see you in the corner over there. Yes, Ebru, go ahead.
Well, hello. Thank you, Leva, for this really interesting talk. I have two questions. I know we're running out of time. So one is, how does low-dose LPS and high-dose LPS affect cholesterol trafficking and storage in the macrophages? I was just curious about that. And the other thing is, what is the contribution of peroxiphagy to inflammasome activation in macrophages?
Yeah, it's a fascinating question, Ebru. So we have some promising lead, but we haven't really checked into this cholesterol trafficking yet. So we suspect that the super low-dose LPS potentially block the peroxisome, block the cholesterol trafficking, cholesterol efflux and things involved in that nature, perhaps through the regulation of SRB and others. So we're gonna further follow up and to check into that. And in terms of inflammasome, that's also interesting. I think the high-dose LPS clearly can induce a driver together with others driving inflammasome. But super low-dose LPS, I don't think that will affect much inflammasome activation.
And what's the contribution of peroxiphagy and the ROS production to?
Yeah, that's right. So we think the peroxisome and paxophagy is also applies in monocyte as well. So we checked in some of the PAX molecules, LPS potently with super low-dose LPS reduce their levels in macrophage monocyte also.
Thank you, Liwei.
Thank you.
Okay, thanks a lot, Liwei. That was great. And thanks everybody for all the questions. So we'll hand it over.
Great. Yes.
Well, so it's a great pleasure to introduce right after Liwei, Jose Fuster from Spain. Jose was best known for his work on the connection between TETs and atherosclerosis. He's now in Spain in the Spanish Institute for Cardiovascular Research and Disease. So I'm just going to hand it over to him to hear what's the latest in his lab. Are you here, Jose?
Right here.
Yeah, there you go.
Okay.
Yeah. So hello, everybody. Thank you for being here. Let me try to share my screen. And well, of course, thank you, Ebru, and the rest of the organizers, not only for the invitation, but also for organizing these great talks that have been so useful during these weird times. I guess you can all see my screen now. Okay, and hear me? Okay. So, well, as Ebru said, I'm going to be talking about clonal hematopoiesis today. And my plan for this talk is to first give you an overall summary of recent research on clonal hematopoiesis and atherosclerosis, including our own contribution to this topic. And then in the last part of the talk, I will try to share with you a little bit of new data. Sorry about that. That has to do with the potential role of clonal hematopoiesis in other cardiometabolic conditions beyond atherosclerosis. But before I get into that, let me just give you a little bit of background on the kind of research that we are doing in the lab. I would say that mostly we are working on aging and the interplay between aging and cardiovascular disease. Sorry, give me just 1 second because I'm having a problem here with Zoom.
Are you good there, Jose? Is it? It looks okay from our end.
Yeah, it's frozen here.
Oh,
Give me just 1 sec. I think I can fix it. Yeah, it's my internet connection. It's done. Now it's good. Yeah. So I was saying that so that mostly we are working on aging and the interplay between aging and cardiovascular disease. Obviously, the rationale for that is that advanced age is the most important cardiovascular risk factor. But we believe that despite that, there is still an incomplete understanding of the connection between aging and atherosclerosis and associated cardiovascular disease. Of course, part of that connection can be explained by the fact that atherosclerosis and cardiovascular disease in general are the result of chronic processes, are the result of accumulative exposure to conventional cardiovascular risk factors. That said, I think that there are epidemiological data that clearly show that there must be something else, some specific age-related cardiovascular risk factors or age-related pathogenic mechanisms that contribute to the development of cardiovascular disease in the elderly. And that's the main topic of research in my group. We have been working a little bit on telomere biology, on cellular senescence too, but probably our biggest contribution, as Ebru mentioned earlier, our biggest contribution to this topic has to do with mutations that lead to clonal hematopoiesis. And as you know, I'm not talking about inherited mutations. I'm talking about acquired mutations, somatic mutations that we acquire as we age, and mutations that for many years we have known that are important in the context of cancer. But now recent evidence show that these somatic mutations probably are relevant in many other age-related conditions beyond cancer. And I think, at least the way I see this, I think it is suggesting that we should change our view of human tissues from a genomic point of view. Because classically, and it makes sense from an intuitive point of view, we believe that all cells in a given tissue, they are the same from a genomic point of view. They share the same genome. They may be different from an epigenomic point of view, transcriptomic point of view, proteomic point of view, but essentially genomically they are the same. However, the emerging reality is that that is not the case, is that most human tissues are actually mosaics from a genomic point of view, because they are a collection of cells that are genomically different because they have acquired different mutations over the years. And importantly, that somatic genome mosaicism, now we know that it happens essentially in every single tissue. And that has been shown in a number of high-profile sequencing studies during the last 5 years. This mosaicism starts already in the embryo, but it's particularly important in the skin, the esophagus, the liver, and particularly in highly proliferative tissues. And I would say that probably if there is a good example of a highly proliferative tissue, that is the hematopoietic system. As you know, the hematopoietic system generates hundreds of billions of cells every single day. That requires a lot of proliferation, and that comes at the cost of a high rate of acquisition of mutations. Indeed, it has been estimated that every single year, each stem cell within the hematopoietic system accumulates at least 17 different mutations. Now, the majority of those mutations, they will be completely irrelevant from a pathophysiological point of view, because either they don't affect phenotypically the mutant cell, or maybe because they are toxic, they lead to cellular senescence or cell death, and that's the end of that mutant cell. But however, recent sequencing studies in humans that have been published over the last 4 to 5 years have shown that it is relatively frequent to acquire mutations that indeed have an important relevance in the pathophysiological setting. Mutations that affect important genes in the context of hematopoiesis, and mutations that provide a competitive advantage to the mutant stem cell, leading to the clonal expansion of that mutant cell. And that clonal expansion is what we define as clonal hematopoiesis, or more specifically, somatic mutation-driven clonal hematopoiesis. And importantly, in a scenario of clonal hematopoiesis, that expansion of the mutant cells initially happens in the bone marrow, but as you can expect, progressively, those mutant cells will also be present in the population of immune cells derived from bone marrow progenitors. So what I'm trying to say is that in an individual that exhibits clonal hematopoiesis, we may end up having a certain number of immune cells, 10, 20%, 30% of immune cells that carry a mutation that has the potential to affect the function of the cell, that has the potential to affect inflammatory responses that are at the center of so many age-related conditions. So this is a phenomenon that could potentially affect age-related conditions where dysfunctional inflammation is important. And that's especially important now that we know that this phenomenon of clonal hematopoiesis is quite frequent. At the very least, I would say that it is present in 20% of individuals older than 60 years old. Specifically, there are at least 20% of individuals older than 60 that carry 4% of mutant blood cells at least. And that has been shown in a number of human sequencing studies in recent years. And overall, the picture that has emerged from these studies, I think can be summarized in this slide. These studies suggest that, as I said, it's quite frequent to acquire one mutation in a hematopoiesis regulatory gene, and that is sufficient to induce the clonal expansion of the mutant stem cell, to induce clonal hematopoiesis. Importantly, these hematopoiesis regulatory genes that are frequently found mutated in human individuals are known tumor suppressors, are known oncogenes important in hematological malignancies. And accordingly, this clonal hematopoiesis associated with mutations in these genes is also associated with a much greater risk of developing hematological malignancies. However, fortunately, these kind of leukemias and hematological abnormalities are rare because they require the addition of multiple mutations that cooperate with the initial mutations. So what I'm trying to say is that the majority of individuals that exhibit clonal hematopoiesis with just one mutation will remain cancer-free, will remain apparently healthy. And that's why this status of clonal hematopoiesis has been defined as CHIP, or clonal hematopoiesis of indeterminate potential. I must say that indeterminate potential doesn't mean that this is a phenomenon that is not important from a pathophysiological point of view, because indeed it was found that clonal hematopoiesis, or CHIP, is associated with increased all-cause mortality. We could think this is because of blood cancer, but that wasn't the case. Indeed, this clonal hematopoiesis is specifically associated with higher cardiovascular disease-related mortality, and more specifically, with increased incidence of atherosclerotic conditions, greater than 2-fold increase in coronary heart disease risk, greater than 2.6-fold increase in the risk of ischemic stroke. These data were published by Sid Jaiswal working with Ben Ebert 6 years ago. And since then, the same group of investigators have validated this connection between somatic mutations and atherosclerotic conditions in other cohorts. Also, there are now new sequencing studies, mostly performed by the group of Stephanie Dimmeler and Andreas Seicher in Germany, that suggest that clonal hematopoiesis can also be relevant in the context of other cardiovascular conditions. But overall, what these studies suggest is that there is an unexpected connection between these somatic mutations that drive clonal hematopoiesis and the development of atherosclerotic cardiovascular disease. However, I always like to remind everyone that these are just associations, and we cannot really make conclusions about causality based on this kind of data. Also, it's not easy to really understand what is going on here at the mechanistic level based on this kind of epidemiological data. For instance, these associations could simply reflect that there is a correlation, that clonal hematopoiesis and cardiovascular disease are two shared but independent manifestations of the aging process. Of course, we always like to think about causality, and it is much more attractive to think that these somatic mutations that drive clonal hematopoiesis contribute to the development of cardiovascular disease. But this association could perhaps reflect the opposite, the other way around. It's possible that cardiovascular disease that is associated with greater oxidative stress, greater DNA damage, greater inflammation, overall cardiovascular disease could somehow facilitate the acquisition of these mutations or the expansion of the mutant clones. So my point here is that we don't really understand the nature and of that connection between cardiovascular disease and somatic mutations that drive clonal hematopoiesis based only on epidemiological data. We need to combine those data with other approaches And that's what we have been working on in recent years. We have been trying to provide experimental evidence that help us understand the nature of the connection between clonal hematopoiesis and atherosclerosis. And in our first contribution to this, we focused our attention on one specific gene, which was TET2. And many times I get asked the question of why TET2. There are different genes that drive clonal hematopoiesis when mutated. Why start working on TET2? And actually, there are many reasons for that. The first one is that TET2 is kind of a cool protein. It's an important epigenetic regulator, that it has many different actions and complex actions. It is an enzyme that it catalyzes the hydroxylation of methyl groups in the DNA, but it can also exert a number of non-catalytic activities. It can, for instance, interact with histone deacetylases and helping the recruitment of histone deacetylases to specific gene promoters. So it was an interesting gene from a biological point of view. And that's one of the reasons why we worked on TET2. But probably the most important reason is that from a historical point of view, TET2 was the first gene that was identified as carrying mutations in cancer-free individuals that exhibited clonal hematopoiesis. And as I said, also, it's one of the most frequently mutated genes in clonal hematopoiesis. In most studies, sequencing studies in humans, you will see that the most frequently mutated gene is DNMT3A, and TET2 is always the second one. And then there are many other genes that are frequently mutated, but at much lower frequencies. So TET2 is one of the top 2 genes of interest. And another aspect that we liked about TET2 was that there are many different mutations that have been identified in this gene. There are no hotspot mutations. There are different mutations that are distributed over the entire gene. And overall are considered to be loss of function mutations. And that was important for us because for other genes that are linked to clonal hematopoiesis, there are hotspot mutations that sometimes act as dominant negative, others act as loss of function. Sometimes we don't even know if it's gain of function or loss of function. But in TET2, it was easier. Overall, it's loss of function, and that made our life less complicated. It made our life easier because we could simply use TET2 deficient, TET2 knockout mice to model human mutations in TET2. So that's what we did. We used TET2 deficient mice, TET2 knockout mice, to generate a mouse model of clonal hematopoiesis driven by mutations in the gene that could help us understand whether these mutations contribute to the development of atherosclerosis. And the strategy that we use for that is quite simple. Essentially, we used LDLR deficient mice, and we performed competitive bone marrow transplantation experiments to generate chimeras that carried only a small percentage of TET2 mutant cells because we wanted to mimic the human scenario of clonal hematopoiesis, where you only have a subset of mutant cells in blood. So we generated specifically chimeras that had 10% TET2 deficient cells. We fed these mice a high cholesterol diet to induce atherosclerosis, and then we monitored the expansion of those TET2 deficient cells, which we could do by flow cytometry because the cells were labeled as CD45.2 positive. And you can see here that indeed there was expansion of TET2 deficient cells. In the white blood cell population, you can see that initially we had 10% TET2 deficient cells, but there was a progressive expansion. At one point, we had almost 60% of TET2 deficient cells in blood. So there was clonal hematopoiesis in this model. There was expansion also in all blood cell lineages, although there was a little bit of bias to generate classical monocytes. TET2 deficient cells tend to generate a little bit more classical monocytes than other lineages. And on the contrary, they generate less T cells. However, I must say that this is a relative expansion because in terms of absolute counts, we didn't see any change in the number of white blood cells or the number of the different white blood cell lineages in blood. So overall, this data that I'm showing in this slide show that our mouse model based on competitive bone marrow transplantations recapitulates 2 essential features of human clonal hematopoiesis. There is expansion of the TET2 mutant cells, but that doesn't affect white blood cell counts or distribution. These mice didn't develop leukemia, so that is consistent with the human scenario of clonal hematopoiesis. Having validated that, the question was, is this affecting the development of atherosclerosis? Indeed, that was the case. We saw that that expansion of TET2 deficient cells was paralleled by a 60% increase in plaque size. And I must also mention that this was done in mice that carried TET2 deficient cells in homozygosity, TET2 homozygous cells. But we also did the same experiment in mice that carried TET2 deficiency in heterozygosity, which is probably more similar to the human scenario, as human mutations are supposed to be in heterozygosity in most individuals. So we did this experiment, and although we saw that TET2 heterozygous cells exhibit slower kinetics of expansion, which you can see here in the gray column, so they expand slowly compared with the TET2 homozygous cells, that is still sufficient to increase plaque size to accelerate atherosclerosis. So overall, this data, in combination with the previous epidemiological studies, allowed us to conclude that there is indeed causality here, and also it also allowed us to establish directionality. Based on this data, we could conclude that somatic mutations in TET2 that drive clonal hematopoiesis indeed contribute to the development of atherosclerosis and associated cardiovascular disease. In parallel, Sid Jaiswal, working with Ben Ebert and Seka Tirejan and Ben Levy, did very similar studies, although using a slightly different approach. And overall, they reached the same conclusion. So it's a robust conclusion that these somatic mutations in TET2 accelerate the development of atherosclerosis. But the other objective that we had when we performed these experiments was to understand the mechanism behind this connection between TET2 mutations and the development of atherosclerosis. And in this context, I must say that we believe that macrophages are key because of a number of reasons, and I'm just going to show a little bit of the data that we generated. If you remember, I mentioned earlier that TET2 deficient cells have a tendency to become classical monocytes in blood. Classical monocytes are the main source of plaque macrophages, bone marrow-derived macrophages. So consistent with that classical monocyte bias of TET2 deficient cells in blood, we also saw that the majority of TET2 deficient cells within the plaque became macrophages, suggesting that macrophages are particularly important in this context. But beyond that, we also found that when we ablate TET2 specifically in myeloid cells, which means mostly macrophages within the plaque, although not exclusively, that was sufficient to accelerate atherosclerosis. So overall, this supported our hypothesis that macrophages are important drivers of this accelerated atherosclerosis. And that led us to hypothesize that this increase in the development of atherosclerosis is mostly due to the generation of a pool or a subset of macrophages, TET2 mutant macrophages that exhibit some kind of pro-inflammatory activity or pro-atherogenic activity. So the next question was to try to understand what is that pro-atherogenic property of the TET2 mutant macrophages. Here we got, when we worked on this, we got a lot of negative data. Our first hypothesis was that it was proliferation, that TET2 deficient cells had a competitive advantage to expand in the bone marrow and also within the plaque. But that wasn't the case because within the plaque, TET2 deficient cells and macrophages specifically proliferated the same than wild-type cells based on Ki67 staining. Also, we didn't see differences in apoptosis, and we didn't see differences in oxidized LDL uptake or macrophage conversion to foam cells or the expression of scavenger receptors and other regulators of lipid trafficking. So overall, this suggested that these are not the mechanisms that make TET2 mutant macrophages more pro-atherogenic. In the end, after quite a lot of work, what we found, and I'm just going to keep a long story simple, what we found is that TET2 deficient macrophages promote atherosclerosis mainly because they overproduce pro-inflammatory cytokines, and specifically because they overproduce IL-1 beta. And we have many evidence supporting that, but we'll just summarize three main lines of evidence that we generated. So for instance, we found that TET2 deficient macrophages exhibit higher transcript levels of IL-1 beta in culture in a variety of conditions. Also in vivo, within the atherosclerotic aorta, we found higher IL-1 beta transcript levels in mice that carried TET2 deficient cells. We also validated this at the protein level, although I'm not showing it here. But not only we found increased expression of IL-1 beta, we also found increased proteolytic processing of IL-1 beta in TET2 deficient macrophages, which, as you know, is a requisite for IL-1 beta secretion. And consistent with this increased cleavage of IL-1 beta, we also found increased secretion of IL-1 beta in TET2 deficient macrophages. which probably has to do with the fact that we also found that TET2 deficient macrophages express higher levels of NLRP3. You all know that NLRP3 is the main component of the NLRP3 inflammasome, this proteolytic complex that catalyzes most of IL-1 beta cleavage within the plaque. So probably this explains the higher proteolytic processing and the higher secretion. So overall, we had multiple lines of evidence suggesting that this increased production of IL-1 beta was potentially driving the accelerated atherosclerosis in conditions of TET2 loss of function. But we also wanted to test that possibility in a more thorough manner. So we went back to our atherosclerosis studies, and we repeated those in vivo experiments in mice that were treated chronically with a specific NLRP3 inflammasome inhibitor, MCC950. Essentially, in these mice, we inhibited NLRP3 inflammasome and therefore NLRP3 inflammasome mediated IL-1 beta secretion. And the results in this experiment were very clear because we found that the accelerated atherosclerosis in conditions of TET2 loss of function was completely suppressed. It was almost reversed in mice that were treated with MCC950, supporting that indeed this inflammasome-mediated production of IL-1 beta is essential for the accelerated atherosclerosis in conditions of clonal hematopoiesis. I must say that we also made a different interpretation of this experiment, which I think is relevant, and I would like to mention it here now, because this inflammasome inhibitor was supposed to be a very potent anti-inflammatory agent, atheroprotective agent. However, you can see here that in wild-type mice, which are shown in white here, this MCC950 only decreased 20% plaque size, and that was not even statistically significant. It was a mild effect. In contrast, in mice that carried TET2 deficient cells, it led to a 50% decrease in plaque size. That was very clear, highly reproducible, and highly statistically significant. So if you see where I'm going here, I think that this data suggests that individuals that have somatic TET2 mutations could get greater atheroprotection from inflammasome inhibition or IL-1 beta neutralization, which is relevant in the context of the CANTOS trial. And I will get back to that in just a couple more slides. Because overall, the data that I showed, I think to summarize it, it provides evidence for causality, and it also suggests a potential mechanism. Our data suggests that clonal hematopoiesis driven by somatic TET2 mutations accelerates atherosclerosis, mostly due to that exacerbated production of IL-1 beta by TET2 mutant macrophages. And you all know that IL-1 beta has many different pro-atherogenic actions in essentially every cell present in the atherosclerotic plaque. In our study, we found a particularly strong effect on endothelial cells that I don't have time to discuss today, but you can find the details in our previous publications or ask me about it later. Because I would like to move on to the main take-home messages from this part of my talk today. The first one, I think it's pretty clear. I think that now we have sufficient epidemiological and experimental evidence supporting that clonal hematopoiesis, at least when driven by somatic mutations in TET2, represents a new cardiovascular risk factor and a new pathogenic mechanism of atherosclerosis that we need to take into account. And second, and also I think this is important, this is a good example of how mechanistic experiments can sometimes provide rationale for the design of personalized treatments or preventive care strategies that are tailored to the effects of specific somatic mutations. And that is what I was mentioning earlier. We found a connection with IL-1 beta that should immediately take us to the results of the CANTOS trial that, as you all know, demonstrated that neutralizing IL-1 beta with canakinumab decreases the risk of ischemic events in high-risk patients. However, it does so at the cost of an increased risk of fatal infections. So I think that one of the messages, one of the ideas that came from CANTOS was that if this kind of IL-1 beta targeted or inflammasome targeted strategies, if these kind of strategies ever make it to the clinical arena, it will be as part of personalized preventive care strategies. In other words, we need to identify the individuals that would get the greater benefit from this kind of drugs. And within this context, our mouse studies were really informative because it suggested that, as I said earlier, individuals with mutations in TET2 should indeed exhibit a superior response to canakinumab. And actually, we proposed that in a review article a few years ago. Back then, it was just a prediction based on mouse data. But in the last couple of years, there have been some human studies that seem to support that prediction. And I would like to very briefly summarize those studies today. So the first study is not published yet. It's work from Alex Beek and Pradeep Natarajan and Sekka Tiresan. They essentially did sequencing studies to identify clonal hematopoiesis in the TopMed whole genome studies. They did sequencing studies in almost 100,000 participants. And what they found was that there was higher levels of IL-1 beta in serum, specifically in carriers of TET2 mutations, not in carriers of other mutations associated with clonal hematopoiesis, suggesting that indeed there is a specific and direct connection between TET2 mutations and a higher production of IL-1 beta. However, this is just a correlation. So actually, it doesn't imply that there is a direct connection between TET2 and IL-1 beta, but there is an easier way of testing that possibility, which is just go back to the CANTOS cohort, sequence TET2, and see if individuals with TET2 mutations indeed exhibit a better response to canakinumab. And actually, Novartis was doing those studies. They presented some of this data a couple of years ago at the AHA meeting. And I would say that the results are preliminary, but very exciting, because they found that canakinumab leads to a 64% relative risk reduction in TET2 mutation carriers, compared to just a 15% relative risk reduction in the general CANTOS cohort. So I think that is huge, because it means that there was a 4-fold greater response to canakinumab in TET2 mutation carriers. I truly think this is exciting, but preliminary. So this was presented 2 years ago, but I'm not sure where this study is now, at what stage is this study. And even if this is validated, I think we need to be careful when we interpret this data, because after all, it's just a post-hoc analysis of a clinical trial that wasn't designed for this. So if we really want to test this possibility, we need to actually do clinical trials to test this hypothesis clinically. Which is going to be challenging. But still, at the very least, it feels like a very nice human validation of our mouse data, and I was happy to see this data. So with that, I think I will finish this part of the talk, and I will move on to some open questions that I think remain unanswered in the context of clonal hematopoiesis. And for some of these questions, I will share a little bit of data with you today. So the first question, I think, it's obvious based on the title of my talk today. We know that somatic TET2 mutations are important in atherosclerosis, but what about other age-related conditions where inflammation is important? And actually, when we look at the previous clonal hematopoiesis studies, there are preliminary evidence that suggests that clonal hematopoiesis can be relevant in different settings beyond atherosclerosis. The connection with heart failure seems pretty strong. We contributed to some experimental studies, and there is also some human sequencing data that supports that clonal hematopoiesis worsens the prognosis and the progression of ischemic heart failure. There is also some evidence linking clonal hematopoiesis to chronic pulmonary disease, type 2 diabetes, and also some neurocognitive disorders where inflammation seems to be important. And today, I would like to share with you a little bit of data, experimental data, that supports a potential connection with type 2 diabetes. And actually, we got to those data when we were trying to answer a much broader question. We wanted to test whether somatic mutations in TET2 could overall accelerate aging. When we tried to answer that question, we realized that the model based on bone marrow transplantations that we generated before, it wasn't a good option for these studies, because when you do a bone marrow transplantation, you need to irradiate the recipient mice, as you know, and that has a number of side effects, GI inflammation, dermatitis, so it's not a good option for long-term aging studies. So we decided to develop an alternative model of clonal hematopoiesis that doesn't require irradiation. And that is what we call an adoptive transfer, which essentially is a non-conditioned bone marrow transplant. Very briefly, it's a simple strategy. We just take recipient mice, we don't irradiate them at all, and we just inject IV a relatively high number of donor bone marrow cells. We do it using a specific regime but essentially that's the procedure. And what we found is that when you do this, again, without any kind of irradiation or conditioning of the mice, you get a little bit of engraftment of the donor cells, between 2 and 3% of engraftment at early time points. But this is stable over time. And importantly, when we inject TET2 deficient cells, this little bit of engraftment is sufficient to allow the expansion of TET2 deficient cells. It takes much longer than in the context of a conventional bone marrow transplantation, but that was ideal for an aging study. And importantly, this strategy has the advantage over traditional BMTs that it doesn't have the side effects of irradiation. And on top of that, it also, it's a better model of native hematopoiesis, which is probably quite different than post-transplant hematopoiesis. So we use this adoptive transfer strategy. Essentially, we generated the chimeras and we just left them in the animal facility for a little bit over 1.5 years. And periodically, we evaluated these mice for a number of parameters. We measured vascular phenotypes by measuring pulse wave velocity. I'm not going to show you the data, but essentially, we found nothing of interest here. We also measured cardiac phenotype. This was done by people in the laboratory of Ken Walls, and actually, we have recently published this cardiac phenotype in these aging experiments. But then we also did some metabolic phenotyping of these mice, and we found something interesting in that context because we found that TET2, the presence and expansion of TET2 deficient cells worsens insulin resistance as mice age. So these are insulin tolerance tests, and I'm sure that you are familiar with this assay. You can see here that at baseline in young mice that have TET2 wild type cells in orange or TET2 knockout cells in blue, essentially they all have the same high insulin sensitivity. By 1 year of age, all the mice have developed some degree of insulin resistance, but there's still no difference between groups. But from that point on, we see that the curves separate progressively because mice that carry TET2 deficient cells develop exacerbated insulin resistance and also a little bit of higher fasting blood glucose levels. And that happens in the absence of any change in body weight or body composition measured by MRI. So this data, we thought it was really exciting because it's suggesting that beyond atherosclerosis, somatic TET2 mutations can contribute to the development of insulin resistance, at least in conditions of aging. We also wanted to test that in conditions of obesity. So we used a mouse model of diet-induced obesity because in that model, insulin resistance developed pretty fast. We didn't use the adoptive transfer. We went back to our previous model of clonal hematopoiesis based on competitive bone marrow transplantations. But essentially, it was very similar to our atherosclerosis studies. We generated the chimeras. We fed them a high-fat, high-sucrose diet. And we found that mice that had TET2 deficient cells also exhibited worse insulin resistance and a little bit higher levels of fasting glucose in blood, suggesting that not only in aging, but also in the context of obesity, the presence and expansion of TET2 mutant cells aggravates insulin resistance. And mechanistically, we also found a connection to IL-1 beta in this context, but specifically in the white adipose tissue and not so much in other metabolic tissues. Both in conditions of aging and in conditions of obesity, we found that mice that carried TET2 deficient cells exhibited higher protein levels of IL-1 beta in the white adipose tissue, suggesting that IL-1 beta could also drive in this exacerbated insulin resistance. So we went back to our animal studies and we performed pharmacological experiments with this inflammasome inhibitor. And again, the results were very clear because we found that mice treated with this inflammatory inhibitor didn't exhibit the accelerated insulin resistance associated with TET2 loss of function. So overall, based on this data, I think we can conclude that beyond atherosclerosis, somatic TET2 mutations can contribute to the development of insulin resistance. And this is a potential indirect mechanism by which these somatic TET2 mutations could contribute to cardiovascular disease, but it also supports a link between somatic TET2 mutations and the development of type 2 diabetes, which is secondary in many patients to insulin resistance. And that made sense, and it was consistent also with some preliminary human studies suggesting that clonal hematopoiesis increases the incidence of type 2 diabetes. Specifically, it was found that clonal hematopoiesis was associated with a 30% relative increase in the incidence of type 2 diabetes. So now we know that there is a connection with atherosclerotic cardiovascular disease, insulin resistance, potentially type 2 diabetes. And I think that we will see that clonal hematopoiesis is probably a shared driver of many age-related conditions. Second question that I think is relevant, I'm not going to show any data here, but I think it was interesting to at least mention it. Okay, now we know that TET2 mutations are important in atherosclerosis, in insulin resistance, but what about other genes that, when mutated, can drive clonal hematopoiesis? That's an important question because TET2 could simply be the tip of the iceberg. There are many other genes that are frequently mutated in human individuals and that are known drivers of clonal hematopoiesis. And for the majority of these genes, we don't really know if those mutations can contribute to the development of cardiometabolic disease. But in addition, I think it's very interesting to mention that there are some human sequencing studies that have evaluated clonality in blood cells. And they found that 50% of the patients, of the individuals that exhibit clonal hematopoiesis have no known driver mutation. In other words, we have no idea what is driving clonal hematopoiesis in those 50% of individuals that exhibit this phenomenon. And I think that's very interesting because it suggests that there are many genes that, when mutated, can contribute to clonal hematopoiesis and remain to be identified. It also suggests that there could be some non-mutational mechanisms of clonal hematopoiesis that also remain to be identified. And most importantly, we have no idea whether this clonal hematopoiesis without a known driver mutation could be associated with cardiovascular disease or other metabolic conditions. And I think that's a question that we should be addressing in the next few years. And then finally, one final question that I think is very important. And this question is, beyond mutations, what else is driving clonal hematopoiesis? And the rationale for that comes from this study that was published 4 years ago that used a high sensitivity sequencing strategy to detect somatic mutations that drive clonal hematopoiesis in healthy middle-aged individuals. So this high sensitivity sequencing strategy allowed these researchers to detect the presence of 1 mutant cells out of 1,000 blood cells. So it was a much higher sensitivity than previous sequencing studies. So when they used this technique, they found that essentially everybody by middle age have mutations in DNMT3A, TET2, or other genes that are supposed to drive clonal hematopoiesis. So if you think about it, this is a little bit in conflict with what I mentioned earlier, that 20% of individuals older than 60 show clonal hematopoiesis. And the difference here is about sensitivity. These 20% of individuals older than 60 that exhibit clonal hematopoiesis, they have a large clone. They have a large number of mutant cells in blood, at least 4% mutant cells in blood. These individuals here, they have a much lower number of mutant cells in blood. So the message that comes from this is that everyone by middle age acquires these mutations at some point. However, only a few individuals show the clonal expansion of the mutant cells, show clonal hematopoiesis. And I think that's very interesting because it suggests that beyond the mutations, there must be something else that drives clonal hematopoiesis, and we don't know what that something else is. There could be specific stressors that facilitate the expansion of those mutant clones. And actually, cardiovascular disease could be one of those stressors. And that takes us back to the issue of directionality that I mentioned earlier. We know that clonal hematopoiesis in some settings contributes to cardiovascular disease, but it's possible that cardiovascular disease contributes to somatic mutagenesis or clonal hematopoiesis, and that's something that we don't know yet. So overall, I think that my message here is that we really need to understand the dynamics of this phenomenon of clonal hematopoiesis, because if we understand the dynamics and the processes that regulate the expansion of mutant cells, we can design strategies to slow down that expansion, or even to reverse the expansion of the mutant clones. And that would be very interesting, not only in cardiovascular disease, maybe also in type 2 diabetes, but also in the context of leukemias and other hematological malignancies. And with that, I think I will finish here just by thanking the members of my lab in CENIC in Madrid, our collaborators and funding agencies, and all of you for your attention. And I will be happy to take your questions now.
Thank you so much, Jose, for a great seminar. Please raise your hands if you have questions, and I'll give the first question to Liwu. Sorry.
Yes.
Go ahead.
Okay. Yeah, nice talk, Jose. So I have two questions. Number one, this TET2 is a known molecule involved in methylation. So any differences in global methylation profiles? Is that mutation, is that gain of function or loss of function? That's question number one. Question number two, we have just done a collaborative study with another group showing that TET2 actually can interact with EGR1. EGR1 is known modulator involved in monocyte differentiation, hematopoiesis. Have you looked at EGR1 connections?
Mm-hmm. So regarding the second question, we didn't look at that, but it's interesting. We should be taking a look at that potential connection. So regarding the first question, to be honest, we didn't do a very thorough epigenetic characterization of macrophages, of TET2 mutant macrophages. Definitely there is less hydroxymethylation in TET2 knockout macrophages. You can expect that. There are broad changes in methylation patterns, but in the specific case of IL-1 beta, what we found is that this increased expression of IL-1 beta in TET2 deficient macrophages seem to be independent of changes in methylation, because it seemed to be independent of the catalytic activity of TET2. We did some experiments with a catalytically dead mutant of TET2, and we still saw an effect in IL-1 beta expression. And the mechanism that we proposed, that to be honest, I think it is only part of the story here, there must be some additional mechanism, was that TET2 was mediating the recruitment of histone deacetylases to the TET2 promoter. So in conditions of TET2 loss of function, there was less recruitment of histone deacetylases, what led to a higher histone acetylation in the IL-1 beta promoter and higher expression of IL-1 beta. Okay.
Zoe, please go ahead.
Great talk. Actually, you almost answered my question, because I'm interested in your inflammasome phenotype. You show that TET2 knockout macrophage not only have increased priming step, and also the complex assembly step. So I'm just questioning about the mechanism of the inflammasome activation. So you show it's on the IL-1 beta promoter, but in terms of the caspase-1 cleavage, so something like the promoter is not going to really explain the mechanism. So my question. The second question is, in addition to the inflammasome activation, IL-1 beta production, so I just wonder why the TET2 knockout macrophage also, drive the other type of the pro-inflammatory cytokine production.
Sorry, can you repeat the second question?
The second question is why the TET2 also, regulate other pro-inflammatory cytokine expression in addition to IL-1 beta.
Mm-hmm. Yeah, that's fine. So regarding the first question, I don't have a good answer. To be honest, so the connection between TET2 deficiency and IL-1 beta overproduction, we see it in every single setting, in every single tissue that we have analyzed, in different models, we see that connection, and we believe that the mechanism is mediated by HDACs, as I mentioned earlier. For the increased expression of NLRP3 in TET2 deficient macrophages, we know it is not histone acetylation, that is not the mechanism that explains that higher expression of NLRP3. We don't think it has to do with the catalytic activity, at least directly. It is not mediated by the catalytic activity of TET2. My guess, and we don't have good data to support that, is that it is an indirect mechanism. That probably TET2 deficiency upregulates the expression of a number of molecules that lead to higher expression of NLRP3. We don't have very conclusive data, but the data that we have suggests that that is the case. And then for other cytokines, we found that in vitro, in cultured cells, TET2 deficient macrophages express higher levels of many different pro-inflammatory cytokines and chemokines, in addition to IL-1 beta, IL-1 alpha, IL-6, a number of chemokines, CXCL1, CXCL2. The mechanism seems to be different for different cytokines. But what I can tell you is that in vivo, the one cytokine that consistently we see upregulated in conditions of TET2 loss of function is always IL-1 beta. and then a little bit IL-6. But many other cytokines that in vitro we see upregulated in conditions of TET2 loss of function, in vivo we don't see them upregulated, at least not to the level of IL-1 beta. So we truly believe that IL-1 beta is the main driver of these phenotypes in conditions of TET2 mutations.
Thank you.
Ada, go ahead.
Hey, Jose, thank you so much. That was fascinating. I have two questions. One is also regarding IL-1 beta, and I was wondering, Andrew Murphy has this paper from a few years ago showing that actually macrophages in the adipose tissue produce S100A8 and A9 that causes the adipocytes to produce IL-1 beta. It goes into the circulation and causes bone marrow cells to proliferate, and then causing monocytosis. So I wonder if you think that the IL-1 beta is working directly on the TET2 knockout stem cells and progenitors, or do you think that these cells produce something else that affect the tissues to produce more IL-1 beta and then make this feed-forward loop?
Okay, so I mean, the question has to do whether there could be maybe a bidirectional connection between IL-1 beta, adipocytes, and then also the hematopoietic system, right? There we have some data that suggests that could be the case, but IL-1 beta seems to be a very small driver of clonal hematopoiesis in our studies. So there have been some studies that have been published showing that inflammation can contribute to the expansion of TET2 deficient cells in the bone marrow. However, that seems to be driven mainly by IL-6, not so much IL-1 beta. Although we also have some data that suggests that IL-1 beta can also contribute to the expansion of TET2 deficient cells. I must say that we have done some studies in obese mice, for instance, and the expansion of TET2 deficient cells in obese mice or lean mice is the same. So I don't think that the adipose tissue is so much affecting the clonal expansion of TET2 deficient cells. And then I'm not sure if you asked also about how IL-1 beta is affecting the adipocytes. Maybe that was part of the question too.
No, I was just wondering if the TET2 knockout cells induce the production of IL-1 beta by other cell types.
That's a good point. Yeah, definitely. So one thing that we know is that IL-1 beta has a lot of autocrine and paracrine actions. So for instance, if we, in the adipose tissue of those mice that have TET2 deficient cells, we have isolated the stromal vascular fraction and adipocyte fractions, and we have also done some co-culture studies with adipocytes and macrophages, and we see that TET2 deficient cells express more IL-1 beta, but those higher levels of IL-1 beta also induce the production of higher IL-1 beta in adipocytes. And that's something that we also saw in the vascular wall. IL-1 beta in macrophages, in TET2 deficient macrophages, is inducing the expression of IL-1 beta in smooth muscle cells, wild-type smooth muscle cells. And that's important because it suggests that having only a small amount of TET2 mutant cells can be sufficient to really establish a feedback loop that potentiates the inflammatory reaction driven by IL-1 beta.
Yeah, and I was also wondering about the hematopoietic compartment in the spleen, in the bone marrow. Why are these cells so good? Are they like better at occupying the niche? Are they better at competing? Like, what's happening?
I mean, essentially for the TET2 mutant cells, there is an effect at the level of self-renewal of the hematopoietic stem cells. So TET2 deficient stem cells have a greater capacity to self-renew. So they expand at the level of the hematopoietic stem cell population. But they also have the capacity to expand in specific progenitors, mostly myeloid and B lymphoid progenitors. But mostly it seems to be self-renewal. We did some studies thinking that it could be cell proliferation, but we don't have very clear data on that. It's mostly self-renewal, I think.
Thank you.
So, well, thank you everyone, and Jose and Liwi for your seminars today. It was really great, and we hope to see you guys next week. We have more coming. So thank you everyone for contributing to the community. Thank you.
Thank you. Thank you so much.
Thank you. Bye.
Bye everyone.